Welcome, everyone. My name is Eamon Hobbs, and I'm the Chairman and CEO of ImmunSYS. Thank you for joining us today for our July 2020 update, where we will share exciting information, including our proof of concept clinical data, which was recently presented at the annual meeting of the American Association of Cancer Research. I'd like to remind you that some of the statements we will make today during today's call will be considered forward-looking statements. Such statements are made based on the current beliefs of ImmunSYS management and are subject to significant risks and uncertainties. If our underlying assumptions prove inaccurate or uncertainties materialize, actual results may differ materially from those set forth in the forward-looking statements. ImmunSYS undertakes no obligation to publicly update any forward-looking statements except as required by applicable law. On the call with me today are Chuck Lick, MD, President and Chief Medical Officer of ImmunSYS, David Crawford, MD, a leading-edge prostate cancer expert and Professor of Urology, University of California, San Diego, and Stephen O'Day, MD, a cancer immunology expert and Executive Director of the John Wayne Cancer Institute and Cancer Clinic at Providence St. John's Health Center in Santa Monica, California. I will now hand it over to Chuck to present our corporate overview. Chuck?
Thanks, Eamon. So we start with some encouraging clinical data that came from a proof of concept study in a group of refractory cancer patients, including a group of prostate cancer patients that demonstrate a 50% complete response rate, really building on initial indications that the systemic administration of checkpoint blockade can effectively treat some prostate cancer patients, but a small minority, and we're excited about that result. It really is what we consider a next generation immunotherapy because it combines both checkpoint blockade, immune stimulation, and a device to create basically an in situ vaccine within the patient. It's very simple to perform. It takes an outpatient procedure by any urologist can do this type of procedure. And we're currently in the process of developing a phase 1b/2 study that will move this to the next sort of level of investigation formally under an IND with the FDA. Next slide, please. We have a very strong and experienced management team that includes a number of people that have great experience in public companies and in taking drugs and drug device combinations forward for approval. Eamon, who you just heard from, has been a CEO serially of 3 different publicly traded companies. Myself, I'm a former public company CEO as well with a lot of strong experience in immuno-oncology and clinical trial development. Joe Girardi, who's been a CFO of publicly traded companies before. John Condra, our Executive Vice President and Chief Scientific Officer, was with Merck Laboratories in the immunology group for 28 years. Augustine Gago, who's our Chief Business Development Officer, again, a lot of public company experience. Marlene Barton, who has a very deep regulatory and compliance officer experience, formerly with Johnson & Johnson. Next slide. Dan Rosanella, who is an expert in medical device development. David Vaughn and Richard Harris, who are both 2 nationally prominent medical physicians, urologists with decades of experience in treating prostate cancer. Lucinda Tennant, who's our Executive Vice President of Clinical Operation, who has run multiple clinical trials and INDs throughout her career. Gabriella Rossi, who's a top immunologist who came out of Steve Rosenberg's group at the NCI. Mario Martino, who has got extensive experience in chemistry, manufacturing, controls for a variety of different types of both biologics and small molecules. Next slide, please. In addition, we have a very strong board of directors. In addition to Eamon and myself, we have Doug Watson, who was the former president and chief executive officer of Novartis. Joe Maroon, who is a clinical professor and vice chairman of neurosurgery at the University of Pittsburgh and formerly was on the board of directors of Mylan Pharmaceuticals. Jeff Cleland, a serial entrepreneur who is currently the executive chair and co-founder of Orpheus Pharmaceuticals. Norm Schwartz, who's the president, chief executive officer, and chairman of the board of Biorad, a $5 billion publicly traded NASDAQ company. Prostate cancer, as many of you know, I think this goes without saying, continues to be a huge clinical problem. especially in patients that recur after local treatment of their disease. The incidence is actually going up, and there's some 33,000 deaths that are projected in 2020 from prostate cancer in the United States. It's essentially one of the largest markets in the cancer area, and unfortunately, the most common malignancy in men. Next, please. One of the challenging aspects of trying to use immunotherapy for prostate cancer is compared to some types of tumors like melanomas or renal cell cancers that are very responsive to immunotherapy, prostate cancer has been a real challenge. There are a few patients that respond, but overall, the majority of the patients do not respond to checkpoint blockade very effectively. The main reason for that scientifically seems to be that prostate cancer are what we characterize as cold tumors, meaning that they don't have any natural lymphocyte infiltration of the tumors. They don't tend to have lymphocytes that are educated against the antigens within the prostate cancer itself. And so they're at the cold end of the spectrum, and there's only a small percentage of patients that are responding with systemic usage of the drugs. So a lot of what this project is about is trying to convert prostate cold tumors into hot tumors by using the tumor tissue itself to create a vaccine within the patient's body. Next slide, please. The way that this is done with the Uravax therapy is cryolysis is done using cold energy to freeze thaw a section of the prostate cancer tumor, which then lyses the tumor contents and antigens within the tumor bed, which are then infused directly with checkpoint blockade and immune stimulating proteins, which then force this material to be distributed up through the lymphatic channels into the regional lymph nodes adjacent to the tumor site. What this does is basically create an autologous vaccine. It's been discovered now that the majority of immune reactions that occur in patients that do have effective immune responses using immunotherapy is that there tends to be one or two single antigens which tend to be very specific to just that patient. And so you really, to get optimal effect, need to use a vaccine derived from the patient. And so when you freeze and thaw a portion of the tumor and basically shatter those tumor cells and release their contents, you're using antigens directly from the patient's own tumor to educate the T cells that are then amplified through the checkpoint blockade and immune stimulation process. Next slide, please. Cryolysis is done only to a portion of the tumor. So as opposed to cryotherapy, where you try to ablate potentially an entire prostate that has been infiltrated by cancer, in this situation, you create a small area of cryolysis where you freeze thaw only a portion of the tumor. That helps leave the rest of the lymphatic drainage intact around the tumor bed. And so right into that same location, the checkpoint blockade molecules and immune stimulatory molecules are infused. So the combination of the cryolysis, intratumoral immunotherapy transport the drugs and the released tumor contents into the draining lymph nodes from that tumor site. And that essentially is creating the in situ autologous vaccine. Next slide, please. So we know a lot of information now about the effectiveness of checkpoint blockade of PD-1 and CTLA-4, two very important immunosuppressive molecules that are present in tumors and immune cells that affect the tumors. GM-CSF is known to be an immune stimulant and has some modest activity on its own. So what we're doing is we're combining all three simultaneously and infusing those directly into the site where the cryolysis occurred to maximize the effect of checkpoint blockade. Next, please. So in essence, if you sort of step back and think about this, what we're doing is using the body's own natural abilities that are enhanced by delivering antigens to create an educated T cell factory so that those antigens are now processed in a situation where CTLA-4, PD-1 are being blocked and GM-CSF is stimulating the immune system simultaneously. And basically, you can sort of think of it that you're creating a T cell factory, T cell expansion within the patient's body, but with a new subset of educated T cells that are tumor specific based on the fact you created an autologous vaccinating effect. Next slide, please. Now, the preliminary proof of concept data, which really needs to be reinforced now with a very rigorous clinical trial under FDA guidance and approval in a phase 1b, phase 2 study that we're currently is being designed by us, really has the potential to remove the current existing ceiling on immunotherapy for prostate cancer. There is data that prostate cancer will respond to immunotherapy. And a study done by Bristol Myers where they combined Opdivo and Yervoy, their anti-CTLA-4 and anti-PD-1 antibodies, systemically in prostate cancer did achieve a 6.7% complete response rate, a 17.5% overall response rate, and 14% of the patients had a PSA decline of more than 50%. It came with a typical type of grade 3 to 5 adverse events related to the immune therapy that is observed with other combinations of Opdivo and Yervoy of just about half the patients. And a number of the patients discontinued therapy. Now, discontinued therapy isn't always a bad thing because it turns out that if you had that much autoimmunity from the drug, some of those patients actually did better. But nonetheless, the side effects are pretty high when it's given systemically, but there is activity. And 17% of the patients responding is actually encouraging. In the proof of concept Euravax study, 50% of the patients had a complete response of the 18 patients that were evaluable in the study of the 21 prostate cancer patients. And the PSA decline of greater than 50% was actually 62% of the total patients. And the side effect profile seems significantly milder than what was observed in the much larger checkpoint 650 study. Since the Euravax study also includes the local delivery of both anti-CTLA-4 and anti-PD-1 antibodies, this is encouraging that local delivery at lower doses maybe allow you to get away with less systemic toxicity. Next slide, please. So based on that, ImmunSYS has undergone developing a very robust plan to develop this. We have to go through the process of having the FDA sign off on all these plans. We've had some initial encouraging discussions with the FDA in a pre-IND meeting. And the idea is to apply for all the appropriate advantages that you can with orphan drug status, potentially going after breakthrough status, potentially designing a phase 2 trial, which is going to have 2 arms, 1 arm for antigen deprivation therapy refractory patients and 1 arm for patients who refuse initial ADT therapy. And ideally, if we get a high enough response rate that could reproduce what we've seen in the proof of concept data, there may be the opportunity in the future to consider accelerated approval. It's early. We need a lot more data to know that that could be the case, but we think that we have very solid clinical design concepts that would be supported by the FDA. In addition, there's some initial data in just a few patients of activity in tumors besides prostate cancer. And we're going to design a second phase 2 trial that will go after looking at cohorts across multiple different malignancies, including pancreatic cancer, non-small cell lung cancer, melanoma, colon, bladder, ovarian, and renal cell cancer. Next slide. We have a terrific scientific advisory board, 2 of the key opinion leaders who you're going to hear from today. Gary Onek was the original person who did the proof of concept work on Euravax, and he's the lead scientific advisor to the company. He heads our scientific advisory board. Neil Shore, who's a nationally and internationally recognized urologist, as is a number of the different, I think all the urologists on our team. Dave Crawford, who will be speaking today, is a professor of urology, University of California, San Diego, really internationally renowned prostate cancer expert with decades of experience in this field. Dr. Stephen O'Day, who's really one of the original pioneers of anti-CTLA-4 and anti-PD-1 therapy nationally, and really one of the world's thought leaders in immunotherapy. Skip Trump, who is recently retired as the director of a large cancer center and has been, again, decades of experience as a prostate cancer expert and opinion leader that many people listen to. Tim Murphy, an expert interventional radiologist, again, of great renown and experience and publication history. Richard Friedhoff, who's a patient advocate and actually a prostate cancer survivor and has worked on other technology fields. George Prendergast, another star in the immuno-oncology world, one of the original developers of endolermin 2,3 dehydrogenase is one of the targets for immunotherapy. So really strong scientific advice that we're getting both in the clinic and from a basic science standpoint from this group. So overall, we think that we're well positioned to build value. It's an innovative therapy. We have very promising, encouraging proof of concept data, experienced leadership. We have a growing intellectual property portfolio with domestic and foreign issued or pending patents that are going to cover both our proprietary molecules, formulations, and devices. A number of near-term milestones, including the intention to get back to the clinic before the end of the year, and obviously a very large unmet need in prostate cancer. With that, we'll turn it over to Dr. Crawford.
Thanks, Chuck. That was a great presentation, and I'm very honored to be with this outstanding leadership and board. As Dr. Link said, my career is focused mostly on prostate cancer, and I've done a lot of research from early detection to treat various treatments and new drugs and things like this. And I say this in meetings, this is one of the most exciting, most promising technologies I've seen in my whole career. Interestingly, listening to Chuck talk about cryolysis and cryotherapy, one of the focuses I've had over the past few years is sort of the male lumpectomy and just treating the area in the prostate that has the cancer, not the whole prostate. And that really boils down to probably about 30 to 40% of men diagnosed. And maybe they could avoid radiation or surgery or things like that, and probably treated nearly 1,000 men right now. One of the interesting things that I saw in some men was that when we treated part of the prostate, we left the rest of it, that there was involution of the prostate over time. It was rare, but it happened in a few of those cases, which means it was some sort of immunological attack on the rest of the prostate by the cryotherapy. And actually, we've had a couple of patients with really advanced small cell of the prostate over the years. We've treated with cryotherapy, chemotherapy, and GM-CSF, which is basically a lethal disease, who have had long-term responses and survival. So, you know, I'm primed when I heard about this, that there's something going on here, and that this mixture of drugs and a way to attack prostate cancer is really a game changer. So my job is to sort of give it a little overview of prostate cancer. And as Chuck said, prostate cancer is the most common cancer diagnosed in American men and second leading cause of death. There's a lot of controversy in prostate cancer, overdiagnosis and overtreatment. And if you look at a lot of cancers like lung cancer, pancreatic cancer, there's almost the number of men and women diagnosed with it are about the same that die. In prostate cancer, there's a great disparity, which is good. 191,000 new cases, 33,000 deaths. So where we want to focus is where the action is and why people die of prostate cancer and what we can do about it. Unfortunately, because of a lot of the controversy about overdiagnosis and overtreatment, a group called the U.S. Services Preventive Task Force has implemented some guidelines that basically say, and their job is to advise family practice docs on screening and various other guidelines, is that don't screen for prostate cancer. Even though we made a big impact in prostate cancer, we were able to have some effect on the disease, but we were overdiagnosing and overtreating prostate cancer. So that's led to an increased rate of advanced disease, and we're going to see more of that. The other thing is that people are living longer with prostate cancer. Men are living longer, so they're at prostate cancer is a cancer associated with getting older. So we're going to see nothing but more cases, and we really need to chip away at what causes people to die and have better treatments. So in this slide, we're talking about this rise in metastatic prostate cancer patients, and that's gone up substantially in the past few years just because we're not out there looking for it. Okay, there are risk factors that are associated with this, and you can see them at the bottom of the slide. Older age, non-Black race, a greater number of bone metastases, PSA doubling time. PSA is probably the most important tumor marker in oncology. It's a great test for finding the disease, following it, and prognosis, particularly things like PSA velocity and PSA doubling time. So can we go to the next slide, please? So, there's a lot in between where we started here about early diagnosis and early treatment to where we are right now, and that's metastatic prostate cancer. So that's the MPCA. The first effective therapy, oral therapy for any cancer, was in prostate cancer back in the 40s, and that was estrogens, and then orchiectomy, bilateral orchiectomy. A guy named Huggins and his colleague Hodges from Chicago later won a Nobel Prize for showing taking men with advanced prostate cancer, altering testosterone, had profound effects on the disease. It didn't cure it. They thought originally it did, but there are 10 to 15% of people with advanced disease that live 10 and 15 years. So I guess we can kind of call that a cure. We also know that that we've come a long way from estrogens and orchiectomy. We have LHR agonists, antagonists, and all different ways to block testosterone available now. And the problem with ADT, or as we call it, two things. One is the fact that it has side effects that we used to think just hot flashes, but now it's hot flashes, weight gain, loss of libido, osteoporosis, mental changes, and it goes on and on. But the big one is cardiovascular, and I'll talk about that in a minute. But it is a great initial treatment. The disadvantages there are resistance, basically, but not in everybody. And when you think about it, most of the cancers we cure are cured by not monotherapy, but by combination. So we don't cure lymphomas with single agent therapy. We use CHOP. We don't cure testis cancer with cisplatin alone. We use cisplatin, bleomycin, Velban, and bleomycin. The same thing with leukemias and so forth. When prostate cancer, has been a slow adaptation of combination therapy. But the biggest one came with under their chemotherapy, and that's with docetaxel. By adding docetaxel to androgen deprivation therapy in men early on and newly diagnosed metastatic disease, the survival rate was increased by way over a year. And then since that, this doublet therapy, and I'll talk about that as I finish up here, has increased substantially. So chemotherapy plays a role. But if you ask a patient, well, what would you rather have, androgen deprivation and chemotherapy or some of these other newer drugs? Most of them will shy away from chemotherapy. I've ran the Southwest Oncology Group GU committee for 28 years, and that is the largest cancer treatment group in the world. And we've done a lot of studies. And actually, the first immunological agent approved in GU cancers was a study we did with BCG in the bladder. But we also did some other studies and then participated in studies with immunotherapy, including the one that was done by Dendreon called IMPACT, which basically looked at this immunotherapy in prostate cancer, the first one approved, sipuleucel T, showing a 4-month survival benefit. Now, one of the things that I did was look at a subset of men in this large trial that were on this IMPACT sipuleucel T trial for castrate-resistant prostate cancer, and showed that when you got patients earlier with their PSA less than 20, 21, the survival benefit was 13 months longer than if you waited until their PSA was over 100. But that's not unusual with cancer. As we know, cancer is a collection of genetic changes. The longer it's around, the more genetic heterogeneity exists and the less responsive it is to therapy. So early is better than later. So let's go to the next slide, please. So prostate cancer likes to go to bone. It's the fertile environment, the seed and soil theory here. And when it is in bone, if you look at some of the x-rays here, you see blastic. That was how prostate cancer presented many times, with pain and x-rays that look like that before we had bone scans. And then we have lytic lesions, which are pretty rare. But that bone is not good bone. And on the left, we see a positive bone scan. What we want to focus on here is with all these new drugs, it's been trying to treat advanced prostate cancer. And you can, talk about what advanced prostate cancer is, but the definition that I've come up with is a cancer that puts a patient at risk of dying. And that can be locally advanced prostate cancer. That can be localized high-grade prostate cancer. It can be metastatic, it can be castrate resistant, a whole bunch of other things. Next slide. So in 2020, we've come a long way from the 40s where we had orchiectomy and estrogens. And now when someone is diagnosed with metastatic HSPC, that means hormone-sensitive prostate cancer, they haven't seen anything. What we've shown, study after studies going back to 2015, is that adding something to androgen deprivation therapy matters. And we've had a blessing of a whole bunch of new drugs in the last couple of years. We've had docetaxel, we've had abiraterone, enzalutamide, apalutamide, radium, and immunotherapy. So we've got all these new drugs for prostate cancer, and it's exciting to have those, which are improving the outcome of men with the disease. Next slide. So this is sort of a timetable, natural history table of prostate cancer, and it's based on the tumor volume and time. And what we see is that a lot of men get a primary local treatment, they're cured. Some of them will fail. They'll get a primary salvage. That's right after the first man. And then there are a group of men we just had a conference yesterday with Prostate Cancer Foundation with a bunch of experts around the country on this thing called oligometastatic disease, which means you have a primary and you have limited amount of metastases, and they respond a lot differently than somebody that has what we call polymetastatic disease, which is a lot of disease. And then as we move through these up and down in this arrow, what we see here is we have first-line treatments for metastatic castrate-resistant disease. It was a lot of those drugs I mentioned, abiraterone, enzalutamide, apalutamide, whatever. And then we have responses. So, this is sort of like the COVID thing. We have the spike, and then we shut it down for a while, but it comes back, and then it keeps coming back with various different aggressiveness, and ultimately the patient dies of the disease. So some of the studies that we're going to talk about and have been done sort of at the end of the rope here with people where they've failed about everything. I think it's exciting to look at, as Chuck mentioned, that one of the phase 2 trials will be people that don't want ADT and don't want the other treatments to go with it, and have this to treat their metastatic disease. I think that this is exciting as it gets. Next slide. So one of the things that really has come up in some work we've done with this and published a number of articles on the cardiovascular morbidity of GnRH agonists and antagonists. And not to spend a lot of time on it, but what we know is that that men on ADT have an increased rate of cardiovascular events, and if you have pre-existing cardiovascular disease, that almost doubles. That's becoming an issue, and particularly older men with prostate cancer, things like that. When I first started treating men with ADT, men in their 70s and their 80s, and their disease would do very well, and a year and a half later they didn't come back, and I found out from their wife that he had a heart attack and died. And we go, well, that happens to older people. Well, it wasn't until about 5 or 6 years ago that it was brought forward that is an issue and it's probably related to a number of things, including GnRH receptors and FSH levels. And I've worked with the guy that won the Nobel Prize for prostate cancer with LHRH, and we have written a couple of papers on that. Andrew Shalley, who is very close to where ImmunSYS is in Fort Lauderdale. He's down at University of Miami. So it's an area where if we can eliminate this or provide options to men, I think it's pretty exciting. Next slide. So the MNCRPC treatment. So now we're talking about CRPC, castrate-resistant prostate cancer. That means that they've had hormone therapy and they have metastases. And we have a whole bunch of things out there. We got doublets, and I've talked about some of those in the pre-castrate-resistant stage. So that's before they've had hormone therapy. Now we're talking about after hormone therapy and failing. And we could spend 2 hours talking about all these studies. Those, the Latitude study and Enzymit just came out, and Titan, and there's a whole bunch of neat new things that are out there. But what we know is that we've made some impact. But let's capitalize on what we have and try to put things together and make it different. Next slide. So the moving into immunotherapy. On the left, you see impact study. That's the one I mentioned, the Dendrion study with sipuleucel T. Basically, that's 3 injections after apheresis. Immunoconjugate, that's injected back into the patient in a very quick IV infusion. And then this does stimulate T cells and so forth. And it's a great therapy. It's well tolerated. And response rates are not terrific, and PSAs don't go down, and I think that's across the board with a lot of immunotherapies, as you can see, at least the early ones. Toxicity there, discontinued, less than 1%. This is a great treatment, but why do we use it? Well, because it improves survival rate. And but it didn't, a lot of the things we look at, PSA decreases and so forth, it didn't. So actually the FDA does not really look at PSA responses as really a surrogate here. The doubling time probably plays a role and other things like that to look at it. But no, ipilimumab, Chuck mentioned that, and Dr. Day will talk about that. And this group is a lot more knowledgeable than I am about some of these things, but it is something that works, and it has some toxicity. And Keynote and Checkmate were other studies that were done in prostate cancer, and these are in men that have castrate-resistant disease that are failing things. What is clear to me here, if you look at grade 3 and 4 events, is that they increase a lot when you start adding doublets and things like that, and as does discontinuation of therapy. But you see some more responses. What's missing from a lot of these is what's the delta in survival, and what does that all mean? And but I think that what this does is at least that it points out that there is some activity to these immunologic checkpoint inhibitors and anti-CTLA-4 drugs, ipilimumab. And so it does make sense to consider those. Next slide. So guidelines. Guidelines sort of make or break things. And we had the head of the NCCN-GU guidelines at our International Prostate Cancer meeting here in Denver last January. And one of the things that he said is that, and I think if you follow the guidelines 100% of the time, you're a bad physician. If you don't follow them greater than 70% of the time, you're probably a bad physician. Guidelines are not rules. They're ways to look at patients and treat them, but there's exceptions to the rules. I meant the guidelines initially did not say you should do targeted focal therapy or male lumpectomy. Right now, that's one of the hottest things around right now. The same thing when I think about that patient with small cell. The guidelines didn't help us very much, but they had, you give platinum and cross your fingers and pray and things might work. But jumping out and doing things and try to individualize care, I think is very important. So what we have here basically is when you have castrate naive disease, so that's patients with metastatic untreated disease, that it's probably not acceptable anymore to use ADT alone. You really need to add, as it says in that big red box, docetaxel, abiraterone, all these other things here to look at. Immunotherapy has not been looked at here, although Dendrion's doing some very interesting studies with men on active surveillance, or doing studies with men who are biochemical failures. And interestingly, when you give this immunological drug, and I'm sure that this will be looked at with this combination later in the residual tumor, but in men that got high risk men that got Provenge, syphilisal T, then had a radical prostatectomy. When you took the prostate out, you could see a zone of inflammation and tumor infiltrates, presumably T cells and things like that, around where the tumor was. So that, I think that's very interesting. So next slide. Just to finish up here, the other big thing right now is germline testing and BRCA1 and 2 and things like that, and identifying people for some of the new drugs, the PARP inhibitors and immunotherapy, things like that. The mismatch, satellite instability, Lynch syndrome, all those things. We're finding out that, and the recommendation is that all men with metastatic castrate-resistant disease have germline somatic testing done. Same way with newly diagnosed metastatic disease. Younger men with high-risk cancers, same thing there. Men on active surveillance probably should too. So next slide. And I believe this is my last slide. Sort of a summary. The bullets that aren't on here is prostate cancer is the most common cancer in men. That's not going away. We're going to see more cases of it, primarily because men are living longer, and prostate cancer is a cancer associated with getting older. If you take the prostates out of 100 men age 80, probably 80% of them will have microscopic prostate cancer and wouldn't have been known in their lifetime. So it is a common cancer. What we have to do is drill down to the 76,000 men who have incurable disease. We have to provide new treatments. We want to improve the overall survival rate. ADT is a great treatment, that works, and it works very variable periods of time. And some men it's out 10 years. We know that certain combinations with chemo with ADT or after ADT and all these new drugs and immunotherapy has some very exciting things. I showed you a couple slides on the positive signal with combination checkpoint inhibitors. And that's what's being done and looked at right here. And is a rationale for that. So with that, I thank you for your time, and I will end.
Great. I'm up, I think, right? So hi, I'm Steve O'Day. I'm the Executive Director of the John Wayne Cancer Institute and Cancer Clinic at Providence St. John's Health Center in Santa Monica. I'm a melanoma oncologist that's been in the trenches for 25 years with immunotherapy, and was fortunate enough to be a small group of physicians, part of a small group that actually introduced ipilimumab, or CTLA-4 blockade, in first-in-human studies in about 2000, and that culminated in melanoma in the first randomized placebo-controlled trial showing a survival benefit in advanced melanoma. And that was 2010, a plenary session at ASCO. And I'll never forget that day. It really launched, I think, the new checkpoint immunotherapy revolution. And I've been intensely interested. Obviously, PD-1 has come along and been involved in those and combinations of CTLA-4 and PD-1. And I have an intense interest now in clinical research, not only in melanoma, but really applying these systemic therapies, combination immunotherapies across solid tumors. So I guess the first question is, why am I interested in this technology? And Eamon called me, and and I've been involved with him in the past. And I've really spent some time thinking about this and reviewing the data. I'm not part of the clinical trial that was presented at ACR, which I'm going to talk about, but I have had the opportunity as an advisor to really go into some deep dive with these case reports. And I'm impressed with the proof of concept of these dramatic responses, and as well as really the technology. And I want to share a little bit of why I'm interested. So this is the abstract that we're going to be talking about that was presented at ACR. This was a group of 27 consecutive patients treated by a 2-physician private practice. But before I get into that, I really want to spend time a little bit on this slide, the mechanisms. Let's go one forward there. So, I think what's interesting about this technology is, there's been plenty of low-hanging fruit with systemic therapy in what we call hot tumors, melanoma being probably the primary one. But there's subgroups of many other solid tumors that are hot. By that, I mean they're very immunologically visible to the immune system and with neoantigens that provoke immune response, particularly an adaptive T-cell response. And so in those hot tumors, it's really about driving both from CTLA-4 in lower doses, but also the primary PD-1 to allow those exhausted T-cells that are already trying to do their job to be reinvigorated and eradicate tumors. So it's very clear that across a number of solid tumors, there are hot tumors that are very immunogenic. They tend to be tumors that have had environmental exposures, high mutational burdens, they upregulate PD-L1, and they need a little help. And we're doing a great job with systemic treatment for those subgroups. We've now, the platform is really combining PD-1 and CTLA-4, because we really know that they're very different checkpoints. CTLA-4 is all about priming and developing an immune response and inhibiting regulatory T cells. And PD-1 is really about resurrecting exhausted T cells. So they combine beautifully together. And it turns out that even though toxicity is very high systemically with the combination, by using lower doses of CTLA-4 at more prolonged intervals, we're reducing some of that toxicity. But it's still considerable. And it's a challenge for oncologists to manage who may not have as much experience. But let's talk about cold tumors, because the prostate is a really good example of a tumor that's pretty cold. And when I think of cold, I think of PD-1 not working well, because there really aren't lymphocytes that are infiltrating and even exhausted. There's really not adequate recognition. If you look at the data with PD-1 monotherapy, as was shown earlier in prostate cancer, really very little responses. It makes sense because there's nothing to resurrect. There's no exhausted T cells to resurrect. There's a primary failure of recognition as foreign. And a lot of that has to do, in my mind, with tumors' defense mechanisms. They downregulate critical antigens, MHC molecules, all the things that make them visible to the immune system, they can downregulate. Each tumor type does it differently. But so when you have cold tumors, it's very interesting that CTLA-4, there generally is some hint that is better than PD-1 when you give it systemically. And that's because CTLA-4 is all about generating that new army of prime T cells that then, although low response rates, there's some response rates. So that's been one problem in terms of cold tumors, is how do you combine systemic treatments where the more toxic drug like CTLA-4 is the better drug, but we still, even with a fresh army of T cells, these tumors, these cold tumors defend themselves well. So that's led to this whole notion of you have to physically or chemically or through radiation perturb that tumor, not the whole tumor, but parts of the tumor, to allow it to become more vulnerable to the immune attack. And this model now, whether it's chemotherapy, whether it's focused radiation, whether it's targeted therapy, short bursts of treatment against the tumor in conjunction with systemic treatment is very exciting. What's interesting about this technology, though, is we may not have to give all that systemic treatment if we deliver the cocktail of cytokines, which is CTLA-4, as well as PD-1, and I didn't mention GM-CSF, which is a dendritic cell or antigen-presenting cell activator. So what you're doing with this particular cocktail is you're activating dendritic cells or antigen-presenting cells. You're activating, priming new early T cells through CTLA-4 blockade, and any cells that get exhausted en route, you're also activating them with PD-1. But most importantly, you're activating them theoretically through their regional nodal presentation, which is an earlier, more robust immune response than late resurrection in the tumor microenvironment. So how you perturb the tumor matters. We know that certain chemotherapy drugs cause more immunologic or necrotic death. We know certain radiation dosing and schedule may lead to different types of cell death. And in this cryogenic model, what interested me was that this lysis basically is a necrotic cell death, which obviously is immunologic. And it matters how we perturb the tumor. And then obviously this creates, once a tumor is perturbed, danger signals are up. And now you're infusing a cocktail that helps activate those dendritic cells that are going to present those antigens. Now the tumor has basically released and revealed its critical antigens. The GM-CSF is activating those dendritic cells to present the antigens. CTLA-4, as the debris sort of flows into the regional lymph node, is activating that primal, very important response. And PD-1 keeps those T cells theoretically more active longer. So it's a really fascinating cocktail without systemic, but basically you're creating your own vaccine in the tumor and then pushing it along to a regional node, which is very different than trying to treat it in the more advanced microenvironment. So for all those reasons, I found this really fascinating. But of course, the proof's in, can we demonstrate some activity with this approach? Next. So again, it's less important when you have small sample sizes to look at percentages in my mind. It's more important to see, is there really proof of concept of something real happening? And that requires really to review these cases carefully. There's clear caveats when, in terms of a retrospective individual doctor group series, but we did look back, and these were 27 consecutive patients. And then there was very rigorous looking at the imaging prospectively and really trying to assess, is there proof of concept here? So these patients most of them were prostate cancer, 6 had other cancers. They got anywhere from 1 to 3 cycles of this cryosurgical lysis with the injection of this very important state-of-the-art cocktail of CTLA-4, GM-CSF, and PD-1. And then they were given post-injection of this 30 days of GM-CSF, which we know subcutaneously activates dendritic cells as well as T cells. And then we looked at these responses. So let's go to the next. So here's the patient group. Again, they were, as you might expect with a prostate population, average age was 65. They're predominantly prostate patients, but there's nothing really specific. And this is what excites me about this technology. If it's demonstrated prospectively to be as good as this looks, there's nothing specific. It's a cold tumor. We've got lots of cold tumors out there that need help. And what you're basically doing is you're putting a probe in not the whole tumor, but an area of a tumor that can be accessible. And so this opens the door up for well beyond prostate. But prostate's a great model because there is some data that the cytokines given systemically, these checkpoints have activity, particularly CTLA-4. And now you're really going to drive that. So what's also interesting about these patient groups, because of the nature of the referrals to this practice, was we had really a peak on this this naive population, right? Because there are many men, for the reasons Dr. Crawford talked about, really may choose not to get upfront androgen deprivation therapy and or chemotherapy. And so almost half of these patients were naive to treatment. We know that in earlier stages or less treated patients, immunotherapy tends to work a little better. But there was also an equally interesting refractory antigen population. There was one of these refractory patients that we actually didn't confirm for sure whether they were truly refractory, but the other 8 were demonstrated. So this is a nice separation. And the average was 2 cycles per treatment. Next. So again, here's, there were 3 patients who were not evaluable because we didn't have follow-up scans. So now that brings it down to 24 from 27, but a very respective response rate, objective response rate of 42%. And you can see almost all of these were complete responses. And we'll show you some of these imaging. And remember, PSA was not a criteria for RECIST, but these were RECIST 1.1. There was also a fair amount of stable disease, meaning they had subsequent scans that showed stability. So a disease control rate overall that was remarkable in this small group of patients. Next. Oh, so go back one slide, I'm sorry. I just want to comment, these were patients that had predominantly local recurrence, either in the prostate bed or in the pelvis, that had either lymph nodes or soft tissues, but many of these also had bony metastasis. So obviously they had to have a soft tissue or nodal recurrence that was accessible to treatment with the cryoablation. So clearly a subgroup of overall patients, but many of these patients had bony metastasis in addition. Next. And here's, of course, just the prostate group. It turns out the 3 that were not evaluable were in the prostate cancer. So half of these had responded. They were complete responses. There was also a cohort of stable disease. And like all immunotherapy responses, most of these have been quite durable. So this is a characteristic of immune response, obviously. And even though PSA decline is not a part of RECIST, you can see that the soft tissue and nodal disease is resolving, and in more than 50% of cases, the PSA was declining by more than 50%. Next. Now here's the 2, again, very small numbers here, so don't be so concerned about the absolute, there's no differences here in terms of really the 2, but both groups, I think, is what is impressive. Both the ADT naive group as well as the refractory group really are showing these dramatic responses and stable disease and PSA reductions. And so again, proof of concept that something was happening. Remember, these patients were not getting systemic checkpoints during this time, nor were they getting chemotherapy or other than androgen deprivation therapy in the refractory cases. Next. What's also interesting to me, everyone talks about, local therapy and we don't want people to get systemically ill and people worry about systemic treatment toxicity with immuno, but remember, you want some demonstration that you're doing more than having a local effect, meaning there are some systemic adaptive T cells that are being produced. Now, that obviously is the case if you're seeing multiple tumors away from your injection site regress, as we saw with these. But it's also good as proof of concept to show that some of the immune-related toxicities that have been associated with checkpoint drugs were also seen, particularly the endocrine toxicities, which have correlated well with overall long-term survival. So vitiligo, which is not seen here, but is an autoimmune disease, and thyroid, both hypo and hyperthyroidism, suggests that there is active systemic immune responses happening. You just don't want, obviously, major toxicities, but toxicities, when given systemically, have correlated with better long-term survival. So I like the fact that there is some grade 1 and 2 toxicities away from the injected site, suggesting that you have developed some adaptive T cell immunity, and those cells are causing collateral toxicity. These are easily manageable when they're grade 1 and 2. The grade 4 toxicity predominantly were involved of an abscess. This one patient had 4 of the 5 grade 4 AEs were related to an abscess that was directly related to the injection. And then there was one death that was unknown etiology. This was a patient, I'll show you the scans, with an unknown carcinoma widespread, who was actually having a dramatic response and had a sudden cardiac arrest. The patient was obese. We know that obviously obese patients with cancer, and particularly patients that are responding, can have a procoagulative state, and who knows what the details were around this death, but certainly PE or cardiac event may have been the cause here. Next. So I just want to give you some review of just very selected cases here. Obviously, here's one that had metastatic melanoma diagnosed. And so this is a case of somebody that's now, I'm going to show you the hormone refractory situation. Diagnosed in '94, had undergone a radical prostatectomy and radiation, investigational therapy, androgen deprivation therapy, chemotherapy, the whole 9 yards. Had metastatic disease and it had a recurrence in the bladder, pelvic area, abdominal and pelvic lymph nodes, and rectum. And here, these are two on the right represented samples of an obturator node here in red, and then this larger pelvic mass on the right. This patient was quite ill with this pelvic recurrence and received 1 cycle of therapy and had a complete remission. This is a scan on the top from January of 2016, January of 2015, and then this is almost 4.5 years later, which is characteristic of these immune responses. They're very durable with no further treatment. Next. So I'm going to give you another example. This is the patient, unfortunately, that died about a month after therapy, but just the PET scan here is so dramatic, I wanted to share. So this is a widespread unknown primary carcinoma. You can see lots of soft tissue and bony metastasis. One of the soft tissue lesions was placed in the S1. There was a posterior element and vertebral soft tissue area, which was selected as a viable tumor to be injected. But you can see here, just in a very short period of time, this is about a little less than 3 months, you can see a very dramatic change in the PET scan. Again, this wasn't evaluable because PET imaging is not considered evaluable for RECIST criteria, but a pretty dramatic response. Next. And here's one of the first, this is a naive patient, one of the last patients treated, no prior treatment. So this is somebody who chose not to have primary treatment with androgen deprivation therapy, had a large mass in the prostate involving the seminal vesicles. There were also multiple retroperitoneal nodes and bone metastasis. And he received 2 cycles of therapy and had a complete remission. Here is just in November of 2018. This is about 3 months later. In January of 2019, you can see the large dominant mass in the prostate has resolved, as well as the seminal vesicle involvement is now resolved. The bony and lymph node disease has resolved, and the patient remains disease-free up until this point with a suppressed PSA. So those are just 3 examples, one of a hormone refractory patient, one of an unknown primary that had a dramatic response, and here is a hormone naive prostate cancer patient. Here, just the follow-up on that patient, in addition to his primary disease in the prostate, that large, you can see here other sites of disease. Here's the prostate, here's a lymph node, another lymph node, here's a bony metastasis, and you can see resolution on this axonal imaging scan. And he continues to be disease-free. So I hope I've given you a little background on sort of why this area intrigues me. I think the field of immune therapy has undergone a revolution with systemic CTLA-4 blockade. Obviously, low-hanging fruit was melanoma, but now systemic combinations of these 2 drugs have really changed the dynamic and allowed us to not only get the low-hanging fruit, but with combinations have higher response rates. But it's still, with systemic treatment, it's still really affecting hot and warm tumors most. And I think cold tumors, we've got to be more proactive at attacking the tumor tissue, allowing that perturbation of the tumor to make that vulnerable, create its own vaccine, And there's lots of data going on with virolytics injecting the tumor, toll-like receptors, using radiation. But these are in combination with systemic treatments. I think what's novel about this is you're really giving local cytokine checkpoints in the setting of an immunologic cell death from cryoablation and pushing theoretically that debris to a robust lymph node that's being activated and primed. And we clearly are seeing some very impressive distant regressions with this small cohort. So 27 patients, 21 prostate, 2 bladder, colon, pancreas. These are cold tumors. They were treated sequentially between April 2015 and December 2019. And you've seen these results, not only in responses, but disease control, and in a cohort of both naive as well as refractory hormone prostate patients. Obviously, there's opportunities here outside of prostate cancer. And overall, the therapy was very well tolerated. It was relatively limited, just a couple cycles in the majority of patients. Toxicity was present and immunologically based to some degree, which is, to me, reassuring that we're developing some systemic immunity. The first patient remains free of disease now almost 5 years, and this is consistent with dramatic immunologic responses. So I think I'll stop there.
I wanted to just briefly review sort of the upcoming milestones of the company so you guys get a sense of how the clinical development plan is going to go. Our current intention is before the end of the year to start the phase 1B confirmatory study using the proprietary CTLA-4 and PD-1s. The phase 2 study is anticipated to start third quarter of 2021. There's a little gap there because we want to take the data from the phase 1 and use that to fully flush out the best design for the phase 2 study, especially because that might become a more important study than just an interim phase 2 study. The exploratory study is the basket study looking at other different histologies. On the green there, it says BLA accelerated approval. That's speculative and really completely going to be contingent upon what the quality and nature of the data is from the phase 1B and from the phase 2 study. But if in fact we're able to do a reproduction of data that looks in line with the proof of concept study yet to be determined, could allow us to consider that type of a move. If that's not possible, then the phase 3 trial would be expected to start sometime in the second half of 2023. Thank you for your attention. We're going to have time for a few questions. Dr. Crawford, Dr. O'Day, thank you for those really nice presentations you give. We appreciate it. If anyone would like to ask a question, we'd be happy to respond.
Hey, Chuck, this is Dave McDonald. Obviously, interesting combo. How important is the device to the therapy, and do you need a proprietary device, or it can be done off the shelf cryo?
Eamon, why don't you take that question since you're
Certainly. Thanks for the question, Dave. Well, as Dr. O'Day pointed out, the first step is to perturb the tumor and release the cellular contents of an area of the tumor, which then allows them to be interacted with by the immune system. So the device is very important in that if you didn't perturb the tumor and just injected the drugs intratumorally, the effect is minimal. That's been tried, it's been studied many times, and it really didn't produce any sort of remarkable results. So the device is critical. It is the first step. Off-the-shelf devices were used in the proof of concept study, but they're not optimized for cryolysis. They're really optimized for cryosurgical purposes. They're multi-purpose tools that have a broad array of capabilities and are quite can be quite complex to utilize. And we're doing something that is what you might call cryo-light. We're doing cryolysis, not cryoablation. Although we're ablating a portion of a tumor, we're also ablating a portion of the treatment zone where some cells survive because we're trying to create a tissue effect that ruptures cell membranes, the preponderance of cell membranes in the area, but doesn't damage the lymphatic drainage in the area. So we're really trying to get a very specific cryolytic effect, and we're optimizing a cryosurgical system to be able to do that at the touch of literally one button.
Thank you.
Any other questions?
I guess this is Dave again. I've got a follow-up,
Thank you.
if you don't mind.
Now, please go right ahead.
So in terms of interaction with the FDA, I mean, obviously the data is extremely impressive. Kind of what's been their feedback and how is that informing that clinical study program that Chuck was describing?
So the initial feedback from the FDA was what we would call encouraging. And I think that they're supportive of going forward with further development of direct intratumoral injection with checkpoint blockade and GM-CSF simultaneously. They also gave encouraging feedback about allowing in a phase 2 trial to enroll patients who refused initial ADT therapy, which we thought was a very important piece of feedback from them. We currently expect to have the new IND filed here in the fall and then start the trial hopefully shortly after that. So I would say I would characterize it as encouraging and positive. They like the proof of concept. Obviously, when you see encouraging data like we've seen, especially the depth of response, that's not a bridge too far for them to say that that's a good idea to kind of move into further clinical development. But I do think that the idea of the treatment naive patients is one that I wouldn't have initially thought about had I not sort of come to understand that some men, when they are fully informed about all the other side effects from the ADT, do in fact don't want to try it, even though that's, you know, probably the best initial therapy for them to get ADT plus, you know, enzalutamide or one of the other new generation drugs in combination.
And if I missed it in Dr. Crawford's presentation, apologies, but how do you think that breaks out in the clinical study? How many folks, you know, what percentage of patients wouldn't want to use ADT first and go right to this therapy?
I think a lot. And there's another interesting aspect of this, and it's something that we've been doing for a long time, and that's treating the primary in the face of newly diagnosed metastatic disease. In a way, you're sort of doing some of that, but not completely ablating it. So I think if you ask a man that has metastatic prostate cancer, what would they rather have? Long-term ADT, chemotherapy, or some of the other agents versus this. And again, I think we have to be very cautious about routinely recommending this, and we're cautiously optimistic about what we've seen already, that this is going to be a game changer for metastatic prostate cancer if the data that are out there all hold up.
Yeah, I agree with you, Dave. I think the way that we see it from a company standpoint is that we have to be very cautious with this group of patients. There is a select group that, you know, do refuse the other therapy, but the other therapy is established by large-scale randomized trials and proven to be effective. And so, you know, be the exception and not the rule, and we'd be very cautious about making that recommendation. But it's surprising to hear patients who absolutely refuse ADT therapy after they hear the effects of the therapy. So definitely a cohort of patients like that.
A lot of patients that that have that. You know, the intermittent therapy was one thing we used for a while to try to ameliorate some of that side effects of ADT. You know, I agree. I think we have to be very, very careful about being too overly optimistic about this, and and recommending it as an alternative. Nevertheless, I think when you look at, if I've been on IRBs before and lots of external advisory boards and data safety and monitoring committees, you haven't totally burned any bridges by
Right.
doing, which I think is the big game changer. I mean, I could see it like, okay, and I wouldn't have any problem with this with patients because we're doing this a lot as sort of lead-in therapy, where you would say, okay, we're going to do this, and this is if you have a CR, you're going to go down this path, which is observation. If you don't, then we have the option of ADT and the other standard things open, and I don't think they're going to go away.
I have a question.
Here,
Can
Ricky.
you hear me?
Yeah.
Hi.
Yes.
So how many people are likely to be enrolled in that phase 1 trial? Is there likely to be a control group, or is it just that everyone's going to get the treatment and that's fine?
So both in the phase 1 and the phase 2, there won't be a control group. Everyone will get therapy. So we anticipate that the phase 1B portion of the trial will be approximately 12 patients. It depends on what outcomes we see, especially in terms of toxicity in the phase 1 dose escalation portion of the CTLA-4 antibody, which is a first-in-man kind of drug. It's being given now in a systemic trial, but it hasn't been given locally to any humans as yet. The phase 2 trial is anticipated to have 2 arms. One arm will have 70 patients, will be 83 refractory patients who have failed ADT and one of the next generation antigen blocking drugs, either enzalutamide or abiraterone or apalutamide. The other arm will be the arm that allows to enroll up to 70 patients who are patients that refuse initial therapy with ADT, the group that Dr. Crawford was just talking about.
Thank you.
Question?
Yeah, thank you, Chuck. And I'd like to thank everybody for joining today, and especially our 2 key opinion leader presenters. Thank you from everyone on the ImmunSYS team. Even in this unprecedented time, our mission to improve the lives of patients with metastatic cancer will continue to be at the forefront of everything we do. Our strong proof of concept clinical data demonstrates the tremendous potential impact Euravax could have in tough to treat populations, and our focus remains on delivering solutions for cancer patients with great unmet medical need. We remain confident in our outlook for 2020 and beyond as we steadily work towards successfully achieving our near-term milestones, including our adaptive phase 1b/2 clinical trial for patients with metastatic prostate cancer, which is planned to commence in Q4 of this year. As our very experienced multidisciplinary team takes the next critical steps towards regulatory approval, we remain encouraged and look forward to providing updates on the business as we continue to navigate forward. Until then, we hope that you and your family stay safe and healthy. Thank you, everyone.